You ready? Shall we start? Yeah. So welcome back, everyone. Today it's my pleasure to host Filipe De Vadder. If you haven't read the flyer, he's a researcher at the Institute of Functional Genomics in Lyon, which is in France. He's been studying how the gut microbiota regulates the development of the nervous system in the context mostly of malnutrition, especially the enteric nervous system. So that's what he'll be talking about today. I think that he's been dragging a lot of the microbiota people, so I see a lot of new faces. So welcome. If you haven't been joining us yet, you can ask your question in the chat while he's talking, and we'll be answering them at the end of the talk. So thank you, Filipe, and looking forward to hearing you talk. You can now share your slides.
Okay, thanks a lot. So, I think you need to allow them, because I cannot share my screen now.
We're not seeing it yet.
No, but yeah, I cannot share my screen actually, so I think you need to.
You cannot. Okay, let me just make you co-host again. Should be working. Try again.
Now it's working.
Okay.
So good. So can you see it? It's fine. You
yeah.
can hear me, hopefully.
Yeah.
So I'm doing this live broadcast from a referee changing room. I've just refereed a game, so that's why I'm having a tie. It's not because it's super fancy. Thanks a lot, Maureen, for the invitation. I'm very happy to be here for this Sunday talk. Everyone who has joined and make some time for this. So today we'll be talking about gut microbes and gut neurons and interactions between both of these guys. So if we have to start to talk about the gut microbiota, of course, the gut microbiota is something that we hear a lot, especially for the past maybe 10, 15 years. We have been hearing a lot about how awesome the gut microbiota is, but you know, this is just where do we have actually microbes in our gut? Well, it starts actually in the stomach. You have a few microbes in there, but then if you go from the duodenum to the distal part of the intestine, which is the colon and the rectum, this is where you have most microbes in there, especially in the colon. So in the colon, you have about 10 to the 11 or 10 to the 12 microbial cells per gram of tissue. That's quite a lot. And it's not only the time of how many cells are in there, it's also the time of how many species are present. And while in terms of biomass, it's quite a lot. It's not only that, it's mostly in terms of genome. The microbial genome, so the metagenome, which is called the microbiome, is actually about 100 times bigger than our own genome. So that means there's a lot of different possibilities, different actions that can be taken by the microbiota that we are unable to do so. So let's go back a little bit back in time, 19th century, with Louis Pasteur, when he discovered, he was the first one basically doing microbiology, and then he thought about something. He has this conversation in the French Academy of Science, that he thought that maybe we could have some kind of animals or a kind of life form that could be present without any microbes. And he thought that if we had this kind of life form that did not have any microbes, under such conditions, he said, life would have become impossible. Well, Louis Pasteur was unfortunately wrong about it, but he didn't live enough to see the development of all this. And development of all this comes to a part of biology that we can call notobiology, which means the study of controlled microbial environments. Now, it was actually in the beginning of the 20th century that we managed to have the first cohort of germ-free animals. Animals are lack any microbes, either in the gut or anywhere else. It was starting with germ-free guinea pigs. And then in 1946, it was actually in Sweden that they managed to find first attempt to obtain a second generation of germ-free rats. Now, why is this very annoying actually to do? Why did it take so long? Because, well, when rat pups are born, as long as they're inside the uterus, well, their gastrointestinal tract is devoid of microbes. So they are born and then they have microbes. So basically you need to take those pups before they're born, so basically a C-section, take the C-section with the uterus, and then you drip it in bleach, so you keep it germ-free, and then you open that in a germ-free environment. Now, once the pups are born, actually, you need to give them milk and feed them. And that's what's so annoying, because the researchers had to come back to the lab at night to feed the pups so that they would grow. And when we had the first generation of these germ-free rats, then they could breed with them. So they could breed in that condition, and then the progeny would become germ-free. So this is basically what it looked like at the time, and now this is what it looks like today. And basically, you have an environment where you have a depression, so that means that the air is not coming in, everything is isolated, and inside this, you have mice that are living in there and that are completely germ-free. So when I was doing my PhD in Gilles Mithieu's lab in Lyon, we found out that actually the gut microbiota could communicate with the host through gut-brain neural circuits. And that communication involved the production of microbial metabolites, which are actually short-chain fatty acids. And the short-chain fatty acids, they are produced actually when you eat dietary fiber, they are fermented, and then they would trigger gut-brain interactions that would actually drive and control energy homeostasis. And well, this was just, okay, so here's the gut, and then you have the distal part, an organ that is quite distal, which is the brain, but you have nerves such as the vagus nerve that allows communication with this. But actually, as a matter of fact, in the gut, you have something else. It's just that the gut is a very precious organ in terms that it contains its own nervous system, which is called the enteric nervous system. And this own nervous system of the gut is basically lying there where you have all these gut microbes. But there's something quite interesting, is that the gut microbes actually cannot communicate directly with this enteric nervous system because you have a layer of epithelial cells. So that means that there is a signal coming from the gut microbiota, and then that can be detected by some epithelial cells or immune cells or something like that, and then that communication will go to the enteric nervous system. Now, this enteric nervous system is extremely important in the physiology of the system because it will allow especially to regulate some functions such as secretion and absorption, also regulation of blood flow control, and this other very important regulation, which is the regulation of contraction and relaxation of different smooth muscle cell layers of the gut, which will allow basically for the food to go from the mouth to the rectum. Now, once you have that, and with this very actually simple organization, we have ganglionated plexus. Here, one's the submucosal plexus and the other one mesenteric. With this very simple organization and tons of neurons in there, they start talking to each other and they regulate the physiology of the gut. Okay, so there have been actually a lot of different papers showing that the gut microbiota regulates function and physiology of the enteric nervous system. I mean, starting from papers studying germ-free mice, and then, I mean, other papers lately have started looking at what this means. So it's actually an expanding field about this. And just a quick look at what these studies have found. Basically, they have found that the gut microbiota can regulate, I mean, blood glucose regulation through activation of specific neurons, that they can regulate gut motility through neuron and immune cells crosstalk. They can also regulate fat absorption and also regulate neuronal loss and neuronal proliferation in the intestine. So just, yeah, if you look actually at intestine, it's quite a densely innervated organ. So this is a whole mount of the intestine. Basically look at that here. It's just an ileum. So it's a distal part. And you have here all the velocities. And then all these things in red actually correspond to this protein, which is beta-3 tubulin, which is expressed in peripheral neuronal cells. And you see that actually the innervation is going through the different layers, and especially going basically all the way through the epithelium, which allows this organ to be densely innervated and probably have some kind of crosstalk in there with what is here, which is the gut microbiota. Now, when I was doing my postdoc in Frederick Beckett's lab in Gothenburg, we actually found out that these germ-free animals that have no microbiota, well, they actually have very, very low innervation of the gut. So here's actually the same protein, beta-3 tubulin in white. You see that there's basically nothing. But once you take these germ-free mice and then you colonize them with microbiota coming from mice that are the same age, same sex, after only 3 days, you see that it starts, these neuronal processes start to grow, and this actually goes along with the proliferation in something that will change the shape of the intestine. As you can see here, the germ-free animals, actually, they don't look at all like something you would have in a textbook, right? In a textbook, you have something like this, very nice crypts and all, but germ-free mice, it's quite different. And we actually focused on a molecule that is regulated by the gut microbiota in the intestine, which is serotonin. And we found out that when we looked at the nervous system, enteric nervous system of the germ-free animals, and you see that basically serotonin is inside the cells, but you don't have a lot of serotonin networks, as you could see in mice, conventional mice with a microbiota. Now, when you take the germ-free mice and you colonize them with the microbiota coming from these guys, you would see only after 3 days something that looks basically very similar. So there's a regulation of serotonin secretion and serotonin circuits in the enteric nervous system. And we found out that actually these networks would regulate gut motility, and they would lead to maturation of different neural progenitors. So this was published in 2018, and we found that the secretion of serotonin was regulated by the gut microbiota, and whenever this serotonin was secreted, then it would allow maturation of neuronal progenitors. Now, since January 2019, I have been in François Lelier's lab in Lyon, and it's a lab that has actually studied how physiology is affected by the microbial environment, and in a context especially of malnutrition, which is a context where you're devoid of protein, you have very, very low protein, growth is impaired. And the first model in the lab was Drosophila. Now, Drosophila has a very simple microbiota, very few species, and something that they found out was actually when you compare the larvae of germ-free Drosophila with larvae that are monocolonized with these guys, Lactobacillus plantarum, especially the string WJL, they saw that this guy, Lactobacillus plantarum, was able to trigger systemic growth and modulate hormonal signals that would modulate and trigger growth. Actually, it was very interesting because then they did that again, this time using mice in the same bacterial strain, which actually is not present naturally in mice. But they found out that when these germ-free animals were monocolonized with Lactobacillus plantarum, it would actually have basically the same growth phenotype as a mouse with a very complex microbiota. And actually, the presence of this bacteria would moderate the deleterious effect associated to low protein diets. And lastly, what has been done is actually this time not only using monocolonized mice, but using mice that are conventional with a complex microbiota. And this complex microbiota, you add, again, Lactobacillus plantarum, and well, they saw that the presence of this bacteria supports juvenile growth in the context of malnutrition. So this is what I've been focusing on in the past 2 years. And something that was very interesting in this is actually that mice that are malnourished, that are lacking protein, so basically these control mice, they have like 18% protein in their food, but these guys, they only have like 4%. It's very, very low protein. And when you have a 4%, but you add, again, the bacteria, well, you have an increase of proliferation in the gut. So this means that, well, in the context of malnutrition, the presence of this bacterium is regulating growth of the gut and growth in the gut physiology. So we focused on this, and I thought, well, okay, let's look at this, but I want to look at the enteric nervous system. That's what we did. Uh, so yeah, we see that microbiota can regulate proliferation, but well, we can also regulate probably differentiation in the epithelium. Okay, let's look at the enteric nervous system this time. How can malnutrition and the microbial environment impact postnatal physiology of the enteric nervous system? And we know that actually microbiota can impact the enteric nervous system either indirectly, through inflammation, bacterial metabolites, or gut hormones, as I showed. Malnutrition completely changes the composition and the function of the gut microbiota. And there can be some kind of vicious circle because in diseases such as Hirschsprung's disease, we have an absent enteric nervous system. It's a developmental disease. We have it, and the enteric nervous system is absent. Well, this leads to malnutrition because there is a lack of motility, there is a lack of absorption, and then if nothing is done, it can lead to malnutrition. So it's some kind of a vicious circle that is driving all this. And well, this means that actually the enteric nervous system is very important in all this. Now, we know that what we look in the mice are mice in the juvenile phase, because when the mice are born from birth to weaning, this is a part where they are actually fed with a very, very rich diet, which is milk, right? So they are fed milk. Milk is very, very rich, and this corresponds to cellular specification and microbiota diversification. Now, after weaning, weaning is a big, big change in the enteric nervous system. It's a big, big change in food, and it's a big, big change also in the microbiota. So have drastic remodeling and microbiota modification. And the idea is, okay, but if we modulate all these parts after weaning in the juvenile mice with our favorite bacterium, well, can we actually see an effect on the enteric nervous system and the physiology of the enteric nervous system? Now, in order to understand that, I told you we have actually two models. The first model is just basically a mouse, a conventional mouse, or SOPF because the health status, but basically it's a mouse with a complex microbiota with more than 100 different types of bacterial species, and there's something that you don't control, right? It's just mice that you buy from a vendor, you keep them in your animal facility, and well, this is something that you have, you keep them, you breed them, and then you can add there the bacteria. But then in collaboration with BioAster, which is a research institute that is located just next to ours, we thought that we could develop a model with a simplified microbiota that would not be a germ-free animal, because as I showed you before, the germ-free animal is not a very good model, right? The germ-free animal has a lot of things that are not okay. They are right. Their motility is not okay. The development of the intestine is not okay. Development of the enteric nervous system is not okay. So we wanted a model that could recapitulate the phenotype of a complex microbiota, but in a controlled environment. So that means that these mice, we have to keep them in the isolators that I showed you before, but we keep them in the isolators. We have a controlled environment, but it's a very simplified microbiota, which means that actually we can very, very well see the variations in this microbiota. And we thought that actually this model could happen just with 15 strains that come from 7 different families. And this, so we have a simplified and controlled microbiota. And so how did we choose actually these species? Actually, well, we sequenced them the microbiota of different mice. So these are mice with a complex microbiota. And among the most representative species, we chose some, and we also chose some from another model of controlled microbiota. So basically we have all these 15 guys. You give the 15 guys to a first generation, and then actually what's really interesting is that these microbiota actually stays. This simplified microbiota stays along generations, so you don't have to do once. Like you take one shot, these mice, they keep there, you keep them in isolators, and then you have these very simplified microbiota. So we had 3 groups here after weaning. We had a controlled diet, and we had either placebo or malnutrition diet with a placebo, or the malnutrition diet, and this time we added Lactobacillus plantarum. So just to show you here how these mice are growing, when the mice are growing on a breeding diet, classically breeding diet, see, after weaning, they start growing. And what's really interesting is actually that either you have a complex phenotype, complex microbiota or a simplified microbiota, you basically have the same phenotype in terms of growth. And then when you have when you have a malnutrition, you have their mice are stunted. They are very small. They have a very small body weight. They don't grow very well because they only have 4% of protein in their food. But when we added the Lactobacillus plantarum, we had an increase in growth. And we saw how could that actually correlate to phenotypes of the enteric nervous system. Now, in order to study this, we actually studied transit time. And how do we study transit time in mice? It's actually very simple. Transit time, you give a gavage with a solution that contains 1.5% methyl cellulose. That is not metabolized at all by the gut. And then you add carmine red just to make it basically, so you make the solution red. It's not toxic, it's not metabolized. It will just go from the mouth to the other end. So you give it, and I love science because sometimes what you do in your lab is just do a gavage and then take your watch and then just wait for the mouse to poop and to make a nice red poop. And once it makes a nice red poop, you're super happy. You write it down, you write how long it took for the mouse to take a red poop. And then that means basically that the transit was done, and the transit time is actually a proxy for the function of the enteric nervous system because the enteric nervous system is crucial for motility in the gut. So what it looks like if we take these mice, basically the control mice, it takes about 2 hours to have a fecal pellet that goes red fecal pellet. But when they are malnourished, when mice are malnourished, it takes much longer, right? So you can actually stay quite a long time waiting for the red pellet, and you can just sing or make noises or anything, and you just not crap, and you just wait. But at some time they will do it. It's good. You write down the time, and then you add Lactobacillus plantarum, and the time is reduced, which is really interesting for us because that means that actually it triggers some kind of activity in the enteric nervous system. So basically, then what we did is that we tried to look at this in a microscopic way, which means that you take the intestine, you remove the mucosa, submucosa, you just keep the longitudinal muscle with the myenteric plexus, and then you look at this in the microscope. It gives you very nice images. So this is just a very small part of the image. So here, all in red, in green is total, so it's a pan-neuronal marker, and then in red you have a specific type of neurons, which are neurons that are nitrogen neurons to express NOS1, which are they are inhibitory motor neurons. It means that when they fire, they basically slow down transit. And what's really interesting is that they slow down transit, but in the conditions where the transit is slowed down, you have an increase, and a specific increase in this type of neurons, either in the complex phenotype or in the simplified phenotype. So it's proof of concept that actually our simplified microbiota works, that we can use this microbiota just to check anything that we want on the enteric nervous system. And well, for what's coming next, we will probably be using this because it's we have something that recapitulates the phenotype of the enteric nervous system. We have something where we can shape the microbial environment with only 15 species, so it's very interesting for us, and then we can partially restore the phenotype observed in control mice by adding the Lactobacillus plantarum. So before I finish, of course, I want to thank my former colleagues in Baker Lab. I showed you some results coming from there. Former collaborators and funding agencies, especially so EMBO, which funded my postdoctoral fellowship in Filipe Baker's lab. Now since 2019, François Lelier's lab. All my colleagues, especially Anne Lambert, has done a terrific job. She's great doing all this. Now with Amélie Joly, who's starting her PhD under my supervision a couple of weeks ago, and our collaborators in BioAster, and of course the funding agencies, because without the funding agencies, I would not be able to work. So Fondation des Treilles and the Agence Nationale de la Recherche funded my positions, and since 1st of October, now the CNRS funding my position. And of course, thank you very much for taking your time to be here on a Sunday evening. Thank you for listening to me, and I will be glad to take any questions.
Thank you, Filipe. Very nice. There are already a few questions from Alike. So I'm going to start. Do you think it is a direct effect or via potential species, whether bacterial, viral, fungal, or archaeal? I'm assuming.
So if it's a direct effect of the species, you mean? Sorry, that was your question.
Yeah, it was toward the middle of your talk. So I guess if the lactobacillus is having an effect on the nervous system directly.
Yeah. So actually, we don't think so, because what I showed you, especially regarding the here, this part, right, regarding the proliferation of the intestine, we could do the same with heat-killing bacteria, and we could do the same with only purified cell wall. So no live bacteria or only purified cell wall, and we have the same phenotype. I haven't looked at the internal nervous system of these mice, either when they're heat kill or with the purified cell wall. But the idea with the purified cell wall is actually that this goes through sensing of a particular receptor, which is NOT2. Because actually when we use NOT to knock out mice, the effects were gone. I think that what's probably going on is that there is some kind of sensing of this cell wall. Now, enteric nervous system is not expressing NOT2, so it may be that this is sensed by some immune cells or epithelial cells, and then the epithelial cells that would translate, they would take that message and basically transduce that message to the enteric nervous system somehow. I don't really know how, but yeah, it's probably not directly the bacterium, but it's some component of it, and we think that it's the cell wall.
She's also asking, is this amplified microbiota also the microbiome present on other surfaces, i.e. skin? I'm not sure. You might, I'm not sure if you got that. Did you get that?
The microbiota, sorry, so if the simplified microbiota is
the simplified microbiota also, do you mind clarifying that question, Ulrike? I'm going to read the other question from Caroline while you clarify that, Ulrike. Is that okay? So Caroline is asking, are the damages caused by malnutrition reversible? I'm assuming is the...
Oh, this is actually a very interesting question. So actually, our model is taking on juvenile malnutrition. Some people have actually done malnutrition in adult stage, but that can be reversible, but then you don't look really at growth. Now, for us, it's very interesting to look at growth. And we have actually started a protocol where after the period of malnutrition, we do a refeeding. And we do a refeeding because, of course, I mean, just take imagine that this would be a young human, right? Going through malnutrition with the first thing, of course, not giving a bacteria, it's just giving them food. The problem is that actually, even if you give them food for a prolonged period of time, when they have had a long period of malnutrition after weaning, they are not able to fully recover at least their growth. That's what we have seen. And we know that from other studies that it's not only their growth, but they have some behavioral parameters that are not okay in the long term. And I think that I haven't looked at the enteric nervous system again in this period when we do the refeeding, but it's a lot of these the parameters are not reversible because we are doing it in a time which is after weaning, where basically I think that if you mess up with the physiology of the system, then you're not able to go back to a normal state. And maybe we think that if we add Lactobacillus plantarum when we are doing the refeeding, well, it can be better than just giving food.
So Ulrich clarified a question. She was saying, is the simplified microbiota the only microbiota present? I.e., it is the only, is it only in the
okay.
But are you reading her question at the same time?
I can read them now. Actually, I didn't see it, but now I can see I can read them. Yeah, so it's not only, I mean, the simplified microbiota we chose is only from the gut microbiota. So we don't know. Actually, we haven't done any swabs from skin, mouth, vagina, or anything else. So I'm not sure how the microbiota anywhere else is, how that is evolving. Of course, it's quite likely. I mean, the only 15 species are present are those. So, I mean, since mice are living together, you know, they're sharing basically their poop. They're basically grooming each other sometimes. So it's very likely that some of these bacteria will be there. Among the 15 species, there are 3 species that are lactic bacteria. So very likely these will also be found in the vagina, but it's just something that I think we have, because we haven't looked at it precisely.
Weren't you worried, so this is me, about choosing a lactobacillus? Because, you know, it is a bit, I do think it is a bit tricky, because you kind of never know. Well, we know it a little bit better now, but, you know, at first there was this dogma that lactobacillus would make you lose weight, and there was this yogurt that was, I'm not going to say which one because this is going to be on YouTube, but this very famous yogurt that was something like, okay, we have lactobacillus plural in our yogurt, so you can just drink it and you're going to feel better and lose weight. And actually some researchers gave these lactobacillus to little ducks, and they ended up looking like geese because they were like so big, and they ended up like sorting different lactobacillus strain and show that some of them would make you lose weight, but some of them will make you gain weight because they are growth factors. So it is always a bit tricky with those because they really don't impact the gut microbiota in the same way. I think you know that, but it's a bit, it's well known now, better known, better characterized now, I do think.
As I said, this specific strain comes from Drosophila microbiota, and it's actually a strain, so it's really the specific strain WJL. I mean, I didn't show the data, but basically in the science paper in 2016, what they showed was that it was strain-specific effect. So when they gave another strain of the same species, they didn't have the growth-promoting phenotype. Now, the growth-promoting phenotype is acting through secretion, and so inducing secretion of growth hormone and insulin-like growth factor in the tissues. So they are growth-promoting, definitely. And actually, maybe, I mean, they will. We haven't looked at what would happen for if they grow, if they could be bigger or something like that, in a normal, let's say in a normal situation, because we are really interested in malnutrition. But yeah, this is how this bacteria was found isolated. Like, okay, this working in Drosophila. There is a very, very well known pathway how it went in Drosophila, and actually it's quite interesting because it's very well conserved in mice with the secretion of IGF. You have something that is very similar in Drosophila, which are the DILPs, so Drosophila insulin-like peptides, and you have basically the same kind of regulation in there. So this is how this specific bacterium was chosen, but something I didn't actually see and I didn't tell you is that we didn't have any effect actually of this bacterium when we had the simplified microbiota, at least on growth. And one of the hypotheses that we have is that actually these GM15 mice, they have among the 15 species 3 lactobacillus species, and probably these species are actually make them at least partially insensitive to malnutrition. So yeah, we don't really know, but it's definitely at least these bacteria, they are growth promoting. Yeah.
Okay. So there is a question from Leslie. She's saying, did the mouse work?
There was Mathieu before.
Sorry?
I think there was Mathieu.
Oh yeah, okay. Did you measure the effect of bacterial transfers on enterochromaffin neuroendocrine cell activity, e.g. Secretion of serotonin, chromatographin? Sorry if it was addressed at the beginning, I missed the first part.
So actually, Mathieu, I cannot answer your question right now, but hopefully if I come back in a year, that's actually Amélie's PhD project. She will be working on all these enteroendocrine cells, which we think are gates from the gut microbiota to the enteric nervous system. So let's see in 1 year, something like that.
Are you already registering for season 2?
Yeah, definitely.
Okay, so Leslie now, did the mouse are kept in groups or were isolated in order to avoid microbes exchange while socializing?
So we always keep mice in cages or of about 5 or 6. So we keep them as litter mates. So they are born together. Basically, what you do when you have mice is that you have two females and one male. You mate them, and then as soon as the female is pregnant, you take the male out, and then you just keep the dams with their pups for as long as possible. And then we kept the pups together when they were juveniles and adults. So they do have exchanges in microbes, but at least in the GM15 model, I mean, the only 15 species that are present are those. And we managed to find that in the longitudinal way that there was the same composition at least for 4 generations. So it's a great model for that, at least.
So Maddie is asking if juvenile animals have access to designated diet ad lib versus scavenge. Do they consume different amounts, suggesting differential CNS effects or regulation?
Well, so what I didn't say is actually that the diets, they are isocaloric. They have the same amount of calories, whether they are low protein or control diet. The only difference is that you compensate the loss of protein by extra carbohydrates. And actually, it goes quite well with what has been described by Stephen Simpson in Australia, who has been studying a lot of these nutritional behaviors in the lack of protein and the role of protein. These mice, they eat more. Now, why do they eat more? Because they probably need to reach what is called a protein level, and they need to reach that plateau of protein. So in order to have the same amount of protein as a control, they would eat more. But I don't know if it's really CNS regulated. I mean, I haven't really looked at that, but they do eat more for sure, and they eat more, but they don't grow more. It's very interesting.
Oh, good. So I think that was the last one. If you're writing a question at the moment, please raise your hand so I don't hang up on you. No hands. Okay, I think that was the last one. Thank you so much, Filipe. I guess see you next year.
Thanks a lot, Maureen. For sure.
Well, okay, so that will be on YouTube.
Maybe not me, but I will ask Emily to come to present her studies for Mathieu for sure. Thanks a lot.
Yeah, okay. Yeah, so I was saying this will be on YouTube, maybe not immediately, but this definitely will. And this will be also on SoundCloud as a podcast. Okay, bye. So there will be no talk next week. See you all in 2 weeks. Bye, everyone.
Bye-bye.
Bye, Filipe.